thank you. Thank you for invitation. And then it's so honored to have uh, this webinar. So this data was already shown in ASHS last year after I got the travel grant from PP Systems, but I redid the experiment, so data will be a little bit different, but same concept research. So today we're going to talk about the photosynthetic efficiency and leaf gas exchange responses of lettuce to both temperature and light intensities. Okay. So before I start my research presentation, I just want to start with my background. So I did my master's degree at Korea University, and my research focused on optimizing automated irrigation strategies. So just I wanted to mention about the uh, history of using CIRAS-3 since 2000, almost 16. So I use that machine to measure gas exchange responses to water status. And then I mainly use one-time measurement and light response curve. And then I started my PhD program at the University of Georgia, still in progress. And then my research mainly focusing on developing supplemental lighting control system. And then I focusing on measuring photosynthetic responses to various environmental parameters such as temperature and light intensity, the data that we're going to talk about today. And then I mainly measure one-time measurement and ACI curve these days. And then we purchased CIRAS-4 in my lab recently, so I use both CIRAS-3 and 4. And then I also want to say thankful that I could get the Trevor grant last year from PP Systems because I really enjoy using CIRAS 3 and 4, and then that helps my research a lot. And then yeah, today we are talking about the supplemental lighting. So as you all know, supplemental lighting is very important to increase crop yield and quality. Of course, year-round production is also available because of the supplemental lighting, especially in the northern area. But always the problem is the operating cost. Up to 30% of the total operating cost can be used because of the supplemental lighting. So we have to develop precise and efficient lighting control strategies so that we can obtain profitability and sustainability. Then how do we control supplemental lighting? The most easy and simple method, we're going to be timer based. So for example, growers can easily set the timer, for example, turn on like 5 a.m. till 7 a.m. and then turn off during the day and then turn on again at the late afternoon, something like that. And there's also a little bit more advanced supplemental lighting control system, which is sunlight based. So for example, PPFD based, if you set the set point as 400 PPFD, for example, and then when the sunlight level is below that threshold, then the supplemental lighting will be turned on. Instead, we can also use DLI base, which is daily light interval, which is a little bit more advanced lighting control system because it will allow you to obtain the minimum requirement of DLI during the day based on the calculation. However, not only light environment will affect the photosynthesis performance. Instead, crop type, growth stage, environmental parameters such as BFD, temperature, humidity, irrigation, CO2 level, all of these parameters will affect the photosynthesis performance. Especially in greenhouses, there's a typical diurnal changes. Sunlight and temperature goes up during the day and goes down. So that is why we have to consider not only sunlight, but also other environmental factors when we control the supplemental lighting. So we will talk about the biggest two environmental factors that affect the photosynthesis, which is light intensity and temperature. So first of all, then we have to talk about the parameter, which is called quantum yield of photosystem 2. So as you can see in the graph, when the P50 goes up, and then the quantum yield goes down. And then quantum yield, the definition of it is fraction of observed photons being used in photochemistry, which means how percentage of light energy is used by plants for the photosynthesis. So when the excessive light intensity is provided to plants, and then almost half of the light energy will not be used for the photosynthesis. Rather than that, it will be dissipated as heat. So it can easily be regarded as light use efficiency. So plants can use light more efficiently on their lower P50. So that is why P50 or DLI-based lighting control system, we use that knowledge. So we growers will want to provide supplemental lighting when sunlight level is low. And how about temperature? Temperature also affects the photosynthesis significantly. So low temperature, high temperature, they all have different mechanism. For example, low temperature will slow down electron transport and reduce silicon member fluidity. And then excessively high temperature will destabilize proteins and reduce repair mechanism. And these all factors will produce ROS, eventually damage to the photosystem, and then reducing photosynthetic efficiency. Now I want to look up what's going on in greenhouse. So we talked about how the light intensity and temperature affects the photosynthesis, but in greenhouse, it's more dynamic and more complicated. So this is a real data that I received from the greenhouse that we have in Athens, Georgia, where the University of Georgia locates. So as you can see, even the sunlight level is same in the early morning and afternoon, but the temperature can be significantly different. For example, summer, the afternoon temperature would be too high, for example, 38 degrees C in August in Georgia. In the winter, the morning temperature would be too low, which is 15 degrees C, lower than afternoon, almost 12 degrees C. It might be too cold for specific crop, and it might be too hot in summer afternoon for specific crop. So that is why we have to consider both temperature and light intensity when we control the supplemental lighting intensity in greenhouse, especially. So I made hypothesis, and then there are two studies, study 1 and study 2. So first of all, photosynthetic performance under constant light and temperature. And then I made hypothesis saying that plants will show constant physiological responses over time because the light and temperature was maintained constantly. And then light intensity will affect photosynthetic performance more than temperature. Lastly, photosynthetic performance will correlate with biomass accumulation. And then the next study, I wanted to see how the photosynthetic efficiency will be affected by fluctuating condition instead of just constant, which is more probably happen in greenhouse. So first hypothesis is plants will use light in different efficiency depending on both temperature and light intensity. And lastly, I want to mention about how we're going to use this knowledge, how we're going to use the effect of light and temperature effects on photosynthetic efficiency. So chlorophyll fluorescence-based biofeedback system, this is my dissertation subject. This system can control the artificial light intensity based on plants' physiological responses. So this system can consider both temperature and light intensity, and also any humidity or any other parameters to control the supplemental lighting. For the study 1, the experimental design is like this. There are 3 different temperature ranges, 18, 25, and 32 degrees C, which is low, medium, and high. And light intensity also low and high, 2 different ranges, 150 and 500 P50. And then these treatments combinations were maintained for 7 days, 1 week. And then this experiment was conducted in gas chamber with dimmable LED and split block design. So for example, if the 30 degrees C was maintained, and then there's a reflectable wall in between so that we can provide 2 different light intensity, which is not disrupted each other. 16 hours of for a period was used, and then lettuce green towers were used. And then those green tower seedlings were grown in the indoor production system. And then the temperature was maintained 25 and light intensity was 250, which is in the middle of the range that I set as a treatment. And then I would like to explain a little bit more about chlorophyllose. I know some of you might know, some of you might be a little bit confused by this concept because this data is very important in my research. So chlorophyllose is re-emitted light by chlorophyll. So when the leaves are provided by sunlight, and then some of the light energy will be used as electron transport, which is what we want, used as a photosynthesis. And excessive light will be dissipated as heat because not all light energy can be used for the photosynthesis. And a small fraction left over of light energy will be re-emitted as a chlorophyllose. So using this parameter, we can calculate the photosynthesis performance, or also we can evaluate how much plants got environmental stress. It's relatively easy and rapid to measure, and it's also non-destructive measurement. And continuous measurement is possible using a scheduled program with data logger or also specific computer program so that we can do the real-time monitoring using this chlorophyllose. Minipan was used for the continuous measurement. I measured chlorophyllose every 30 minutes during the photo period, and then every an hour during the night. Uppermost fully expanded leaves were used, and then I measured two different kinds of parameters from the chlorophyllose. First of all, quantum yield of photosystem 2 that we mentioned earlier, which is can be also regarded as light use efficiency in an easy way. And then the next one, maximum quantum yield of photosystem 2. This means the highest potential of plants have they can use the light use efficiency. So we measure the maximum quantum yield of photosystem 2, which is FV of FM during the night after plants were fully dark adapted. Photosynthetic rate and rapid ACI curves were also measured using the CIRAS-3 from PP systems. At the end of the experiment, days after treatment 7. And then, so I would like to mention a little bit more about how to measure the rapid ACI curves. So we set the stepwise program, which is increasing the CO2 level over time. So we measure it without leaf and with leaf, and using the two curves and then recalculate the CI level, and then ACI curve video will be done to get the real curve. So I actually enjoyed this rapid ACI curve system because it takes only around 7 minutes per each step, which means total almost 15 minutes for both empty and leaf measurements. So per each sample, it takes only 15 minutes to make the ACI curve. And then also at the end of the experiment, I measure fresh weight, dry weight, and then sheet water content was calculated, and chlorophyll contents was measured too. And yeah, this is statistical analysis information. So this is the result. For the quantum yield of photosystem 2, which is light use efficiency, as you can see, the biggest difference between light intensity is that under the high light intensity, the plant showed significantly lower quantum yield. That's already anticipated from the graph that I showed earlier because plants cannot use light efficiently under high light intensity. But how about temperature effect? It's a little bit smaller effect compared to light intensity, but still there is difference. So for example, 150 we can see the left-hand side graph, which is 150 p50. 25 and 18 degrees C showed quite similar quantum yield, but 32 degrees C, the quantum yield decreased a little bit at the end of the experiment. But however, under the 500 p50, the quantum yield of 3 all three different temperature conditions, they all increase over time. And also, especially 18 degrees C, it decreased quite a lot. And then they were showing similar quantum melt at the end. That is because of the plant's acclimation process. So plants recovered and acclimated to high light intensity and low temperature under the high P50 condition, so that they show the similar light use efficiency. And then, as you might see, there is a specific pattern, which is low light intensity and high temperature. And then high light intensity and low temperature. That combinations were not good for the light use efficiency, which is quantum melt. That is because plants were naturally exposed to low light and low temperature, and high light and high temperature. But when it's combined in vice versa, which is low light and high temperature, high light and low temperature, that is just not good for the plants to do for the light use efficiency, because of the photo inhibition can occur in these conditions. So this data is the average quantum melt during the whole 7 days. So the temperature effects were a little bit diluted because of the changes over time. So p-value was barely 0.06, but the p50 effects were very strong, because high light intensity decreases the quantum melt. And then there's also interactions between P50 and temperature, which means plants will show different patterns depending on the P50. So which is it's the same the pattern that I mentioned earlier, the high light and the low temperature, and then the low light and high temperature. Those combinations is not good for the quantum melt. That is why there was an interaction between P50 and temperature. But still, the light use efficiency was affected more by light intensity than temperature. This is the maximum quantum melt, which is FE of FM data. So we, generally speaking, below 0.83 indicates environmental stress on photosystem, which is photo inhibition. So as you can see, the line indicates the 0.83. And 25 degrees C, they all showed mostly over 0.83, which means plants were not really got the photo inhibition in the optimal temperature range. But however, 18 and 35 degrees C showed below 0.83, which means they experienced the photo inhibition. But however, they're not just stayed low, especially under 500. They at the maximum quantum melt increased significantly over time, and then became very similar to 25, and then it was almost 0.83, because plants did recover and acclimate to the conditions. Photosynthetic rate data was also measured at the end of the experiment, day 87. Temperature and P50 effects were very significantly different, but a similar trend as quantum melt photosystem 2. But we can see the bigger difference under high light compared to low light. So under the 150 P50, all three temperatures showed similar photosynthesis rate, but under 500, the photosynthetic rate was quite different. This is the ACI curve data. The left-hand side graph is showing 150, and the right-hand side 550 data. So as you can see, the maximum photosynthetic rate that we can achieve in the high CO2 level is much higher under 500 because of the high light intensity. Plants can use the light energy and they use it for the photosynthesis. But when we look up the temperature differences, we can also see the same trend as we saw in the quantum melt and photosynthetic rate data, which is 18 degrees C under low light, which is 150, has high photosynthetic maximum rate. And then in the another side, 500 P50, and then the low temperature 18, it showed the lower photosynthetic maximum rate. So we can also see the same trend. And then chlorophyll contents, we couldn't see much difference in chlorophyll contents from the P50 and temperature differences. I think that is because 7 days were not enough for planes to show a different level of chlorophyll contents. Lastly, this is the biomass result. So fresh weight and dry weight, you might recognize that the patterns are a little bit different from the photosynthesis quantum melt data. So 25 degrees C produced the highest biomass, which is different from the physiological result. My assumption is that there must be multiple different benefits for the 25 degrees C to produce high biomass, not only for the photosynthesis, but also root growth or other nutrition, transportation, for any other, physiological responses can be affected by temperature, not just photosynthesis. So that is why 25 degrees C, which is optimal temperature, produce the highest biomass compared to other low and high temperature. And then high light intensity increased dry weight, but not for the fresh weight. So when we compare the 18 degrees C under 150 and 500, it's both C, and the other temperature conditions also show the same trend under the fresh weight. But the dry weight, the light intensity increased the biomass significantly. That is because there is a difference in sheet water contents. So depending on the experiment and depending on the researcher, sometimes we only see the fresh weight or sometimes we only report dry weight. But there's a different meaning. So if you want to focus more on the production in the reality, because for example, lettuce are still sold in fresh weight instead of dry weight, then we can focus more on fresh weight. But instead, if you're more interested in the physiological responses and how a carbon accumulation occurred under specific conditions, then we can look up the dry weight. But I want to say that sheet water contents is different under the different PFD. So under high light intensity, 500, the planes will have lower water contents and then have more dry weight in the same fresh weight. So we have to consider sheet water contents depending on the research purpose. So for the study one, I want to see if my assumptions was correct, but some of them were not correct. So first of all, I thought the plants really show constant physiological responses over time, but actually it was not, because lettuce plant recovered and acclimated to high light intensity and also low temperature, and then non-optimal temperature conditions over time. And then light intensity was indeed affected by photosynthesis, affect photosynthetic performance more than temperature. But there was a specific pattern, which is low light combined with high temperature or high light intensity combined with low temperature. That combinations were not good for the photosynthetic performance. And then the last hypothesis was also not correct because photosynthetic measurement and physiological responses in terms of photosynthesis was not always correlated with growth because of the other factors might also affect the biomass accumulation among different temperature. Yeah, 25 degrees C condition had additional benefits for biomass accumulations. So this is a study too. It's a little bit shorter, relatively. So this is the experiment design. So it's similar to the graph that I shown, which is the real data from the greenhouse that I received. So I set the summer and winter conditions separately so that as you can see, photo period is longer in the summer conditions and shorter in winter conditions. And then I set the same P50 peak because we usually use shade curtain in summer because of the too high temperature and light intensity. But the temperature ranges in different way, and then the peak is not in the noon. Instead, it's around 3 p.m., which is mimicking the greenhouse conditions. So for the summer, temperature goes up from 25 to 40 as maximum. It can easily happen in Georgia, and then in the winter, temperature ranges is from 10 to 25 degrees C. This experiment was also conducted in growth chamber with lattice with 4 replicates. So this is the photosynthesis efficiency result. Let's look at the left-hand side summer data first. We were going to compare the 2 time points having the same light intensity, which is 250. But however, the temperature was almost 10 degrees C different, even though the light intensity was exactly same. And then the photosynthetic efficiency, which is quantum of the photosystem 2, was quite similar in summer conditions. But however, in the winter, also same 250 P50 showed 12 degrees C difference in the simulation, and then the photosynthetic efficiency was quite different. 0.604 under 13 degrees C and 0.688 under 25 degrees C. And then I think that is because the crop and cultivar specific result, maybe the lettuce that I use has more heat tolerance characteristic, but it's not tolerant to the cold conditions. So the fact that I want to emphasize in this experiment is that we have to consider not only the light intensity, but also the temperature. So for example, if you want to grow cold sensitive crop and then provide the same light amount in the early morning and afternoon, but actually in reality, the plants are not using the light in the same light use efficiency, which is in the morning, plants are not using the supplemental lighting that I provide in a less efficiency. So we will prefer to provide more light, more supplemental lighting in the afternoon instead of morning, and vice versa in summer. If you are trying to grow the heat sensitive plants, then we have to also consider the temperature and light intensity combinations. So this is summary and conclusion. The assumption, the hypothesis was correct, which is the crops specific and cultivar specific response are anticipated, but still light use efficiency were affected by both temperature and light intensity. So for example, we can have like this kind of a strategy depending on the crop and cultivar. So through this experiment, we can think that dynamic and efficient lighting control strategies are required instead of just consider only ambient sunlight levels. So that is why I want to mention about the chlorophyll fluorescence-based biofeedback system, which is my dissertation topic. So this biofeedback system is suggested by Dr. Van Rosen, my previous advisor since 2016. And the system can adjust LED light intensity based on photosynthetic performance. Either quantum field or electron transport rate can be used as threshold. And this concept is very intuitive. Plants controls the light condition. So this is a real picture of the biofeedback system that I'm using. So plants are measured by the fluorimeter, which is measuring the chlorophyll fluorescence. So measure the chlorophyll fluorescence continuously. And then the data is sent to data logger. And the data logger will decide the LED light intensity as much as plants needs. And then it will affect the quantum yield again. So that's why it's called biofeedback. It's like a closed loop. And then we can only provide the light intensity only as much as plants can use in an efficient way. And this system can be used in the vertical form and also greenhouse. Both growing system can be used. So lastly, I want to show the basic biofeedback lighting control logic. This is very simple. So basically, let's say I want to use ETR instead of quantum mil because ETR is which is can also be regarded as overall photosynthetic rate in light reaction. So measure the current ETR and compare with the target ETR. And then when the current ETR is higher, then the light intensity is too high for the planes, and then we can decrease to match to the target ETR. And vice versa, we can decrease it when the current ETR is lower, and then we can increase the light level to match to the current ETR, and then measure the ETR again continuously. This is the end of my presentation. Thank you for my both lab, Dr. Juanita Ferrez's lab, and then Mara Fernandez's lab. And then the study was funded by the LAMP project. And also, I'm really thankful to the PPS system that funded me so that I can present this data last year in the ASHS.
Thank you for attending the webinar. Contact us today to learn more about the CIRAS-3 Portable Photosynthesis System, the fourth-generation portable powerhouse elevating the high-level field research experience worldwide.